Okay, thank you very much. Can everyone, is the mic working okay? Yeah, wonderful. It's an absolute pleasure to be here today. I'm looking forward to taking you on a bit of a journey through plant evolution over the next hour. So let's just jump straight in. So for me, the diversification of plants on land really was one of the major events in Earth history. And when we look back over the past 4.5 billion years of Earth history, we can see that for the first 4 billion years, the terrestrial surface was a pretty inhospitable place to be. However, this all changed roughly 500 million years ago. Plants radiated out of the water onto land, and geologically speaking, in a really short amount of time, they were transformed from single cells to trees. And a real kind of aim for my research has been to try and understand what enabled this remarkable radiation. But let's take a bit of a deeper dive into this block of time. So when I'm talking about this initial radiation of land plants, the period of time, or the era of time I'm really focusing on, is known, geologically speaking, as the Paleozoic. It spanned between 540 million years ago through to 250 million years ago. And just to make sure we're all on the same page, I know most people aren't that familiar with land plant evolution, I'm going to take you through a kind of best of what was happening early during plant evolution. So for me, the story of plant evolution starts at this point, 470 million years ago. These, in some ways, remarkably uninspiring fossils, these tiny masses that we can only examine under an SEM, these tiny little circular structures here, represent some of the earliest plant spores. These were described by Charlie Wellman and colleagues back in 2003, and these represent our earliest direct evidence for plant life on land. So this tells us that plants were present on land at this time, and it tells us that they were producing spores, and these spores were being blown all across the land and into the water as well. So this was at 470 million years ago. But sadly, we have to wait another 40 million years to actually have any idea what the plants were that were producing them. So skip forward to the Silurian period, and 432 million years ago represents a really key time during plant evolution, because this is the earliest time point where we actually are able to find plants that look like plants, and they're termed the body fossils. So in many ways, you might look at this and think, this looks like no type of plant I've ever seen in my life before. It's got no leaves, it's got no roots, it's just a branching stem with these globular structures at its tips, which are its reproductive structures known as sporangia. Now, the other thing to notice is this scale bar on here is only a centimetre tall. So during this period of time, the Earth was kind of dominated by a terrestrial flora that would barely even scrape your ankles. And the amazing paleoartist Richard Bisley has had a go at really capturing what that environment must have looked like. To me, this looks just stranger than science fiction, a world where the entire flora is only a centimetre or so in size. And this was occurring, and we think, widespread during the Silurian period. And we had to wait a little bit longer until we jump over the border into the Devonian period for plant life to really take off and to be the kind of life we know and expect today. So in the early Devonian, when we jump over into this period of time, we find again these strange little branching stems, which are often termed as the genus Cooksonia. But alongside these represent the first plants that I think if you were standing there, you'd say, hey, that looks like a plant. It's got leaves. It's got a complex rooting system. And so this is occurring in the early Devonian. We're finding plants, and not only are we finding plants that look like plants today, but we're finding them almost everywhere we search for them. So huge amounts of geological work across the world has demonstrated that this terrestrial flora was diverse, and it was present if you're searching in Europe, in the US, in South Africa, or in South China. So almost everywhere where we have rocks from this time period, we come across fragments of these plants. And what I like to think as well is actually this is probably the first time in Earth history that if you were looking in on Earth from outside, from space, this would be the first time you might get a little bit of a green tint to the terrestrial surfaces. But what we can be sure about is if there wasn't a green tint at this point in the early Devonian, you didn't have to wait very long until there definitely was. And that is because as we move from the early Devonian to the middle Devonian, we suddenly have the first forests. So this really was a turning point in Earth history. For the first time, the terrestrial surface was covered by a kind of lush forest ecosystem. And I really love this cover. This is the front cover from Nature from 2012, when Bill Stein and colleagues described this earliest complex terrestrial forest from New York State in America. And this really captures the kind of diversity of the plant and animal life present in that forest. And there's been a lot of continued work to try and understand these forests in more detail. This was the first one really described in detail back in 2012, but it's really exciting that we're still discovering what these early forests look like and how extensive they were. In fact, work actually from here at the University of Cambridge, led by Neil Davies, he's over in Earth Sciences, recently, earlier this year, demonstrated the earliest fossil forest is actually preserved here in the UK, in Somerset. It's been overlooked for a long period of time and would have looked something like this. This is an artist's reconstruction of the types of plants that were growing in that forest. And further work by Bill Stein and colleagues on those sites in New York is revealing just how extensive these plants and trees were. This paper from 2019 shows an image taken with a drone of looking down on a rooting system preserved. You can see this is where the central trunk would be, and you can see the roots radiating out in all directions. Now, rather than earlier, where that Cooksonia had a scale bar of 1 centimetre, we're now looking at a scale bar of 1 metre. So suddenly, plants have got huge, and we're recognising these forests. They were covering large parts of the terrestrial surface. And then by the time we reach the end of the Devonian period, where definitely our own tetrapod ancestors were crawling out the water, they would have been crawling out into a forested world, which would have had many of the similarities we'd know today. They would be able to recognise things like seeds. They'd recognise a variety of different ferns, like the horsetails, and of course the club mosses, which are a really ancient lineage of plants I'm going to be talking to you more about today. And so despite the fact this was a very alien world in many senses, those plants were there, and it really set off a trend for the fact that forests would be a key feature of the terrestrial ecosystem all the way through to the present day. Okay, so just to give a very brief recap on that, as I said, for me, I still find this quite remarkable that during this period of time, during the Paleozoic, we go from the first spores to these tiny little alien plants, and then suddenly we have forests which persist all the way through to the present day. And so at the centre of my research is trying to understand this question, which is what enabled plants to explode in their diversity? How do we go from this alien world to one that we find quite familiar today? And over the next few minutes, what I'm looking forward to doing is telling you two stories focused on work we've done in the lab, trying to understand this question in more detail. So today I'm going to be telling you two stories focused around the evolution of leaves. I've worked on a number of different parts of a number of different organs in plants, but I think leaves are in some ways some of the most exciting. We know that any innovation in leaves would enable them to capture light, to photosynthesize more and more diverse in a diverse fashion. And yet there's still so many big questions about when leaves evolved and how they've changed through time. So I'm going to tell two stories about the evolution of leaves, and the second thing I'm going to really highlight is the role of fossils with exceptional preservation. So let's dive in first. We're going to start in the Devonian, that early period of time when plants are just beginning to go from the alien landscape into those early terrestrial ecosystems, to really try and pinpoint when leaves evolved and how they evolved. And to do this, we're going to take a little field trip further north, a little bit further north than where I set off in Edinburgh today, up to the rural heart of Aberdeenshire, to the small village of Rhynie, which is nestled in the heart of the Aberdeenshire countryside. Now, we visit the village of Rhynie today, you're not very likely to come across these fossils which, especially amongst the community of paleontologists, this site is famous for. This is last year, almost around exactly this time last year, we were standing in the field, in the site of scientific special interest in Rhynie itself, a site of special scientific interest where you're far more likely to find sheep grazing than you are to find these exceptional fossils. However, this site has become incredibly important for the evolution of life on land. And the reason for that is about 6 feet below our feet, if you dug down with a digger, you'd come across some bands of rock known as the Rhynie Chert. Now, these bands of hard flint-like rock preserve evidence of an early terrestrial ecosystem. And again, Richard Bisley has captured this in amazing kind of detail of showing what this ecosystem would have looked like. I have to say, very, very different from rural Aberdeenshire today. You would have been in an ecosystem with these giant geysers like Yellowstone National Park or in Iceland. These geysers would be exploding, there'd be bubbling hot springs, and they'd be gradually inundating this rich ecosystem that was growing around the outside of the hot springs. Now, we're very lucky because that water in the hot spring was laden with silica, and as it actually flowed over this terrestrial ecosystem, that silica was deposited. And it was deposited both within the plant cells and around them. So effectively, this entire ecosystem was frozen in time. And therefore, as I said, what is now just a small field in Aberdeenshire has become one of our most remarkable lenses into early life on land. And I think there's three, for me, key reasons why Rhynie is just quite so important. The first is its age. I touched upon that just a minute ago. The period of time when the Rhynie chert was forming is in the early Devonian. It has this perfect thing between the kind of alien world at the beginning here and forests. So it's really capturing a period of time of explosion in plant diversity. So its age is really important. The second thing that's really important about the Rhynie chert fossils is they are preserved in situ, which means where they were growing. Now, if you haven't really given any thought to fossils before, what I think a lot of people forget is the vast majority of fossils that we examine are not preserved where they were actually living at the time. In the case of plants, they fall into streams, they're washed into rivers, washed into the sea. And I think this example really captures this incredibly nicely. It's this remarkable palm frond surrounded by the fossil fish, indicating it's at the bottom of the lake. And so it's really quite unusual to have plants preserved actually where they were growing. And when they're preserved in the place where they're actually growing, we get new insights on their rooting system. We can see in more detail how they're interacting with the soil environment surrounding them. So that's the second thing. And the third thing, which for me makes them so impressive, is that they're preserved with exceptional levels of preservation. And so just to illustrate this, if we go back to that early species I showed you, Cooksonia, the first branching fossil, we find plants that look very similar to this in the Rhynie chert. You can see the same sort of thing, branching stems with these terminal reproductive structures. But the big difference is, in the example here of Cooksonia, if you examine this under a microscope, all you see is a few coaly outlines of where the plant tissues were. You can zoom in on this more and you'll never see any more details. In contrast, when you cut the fossils in the Rhynie chert, you can make thin sections of them and examine them under a microscope. So this is a transverse section through the stem of Rhynia. It's only a couple of millimeters in diameter, and what you can see is you can see individual cells within a plant that grew 407 million years old. You can see the conducting tissue at the center, you can see the epidermis where the stomata were for gas exchange, and you can even see this kind of brown line going round. This is really fascinating. This is evidence of mycorrhizal symbiosis. This is evidence of where the fungi were actually forming a symbiosis with the plant. This is all frozen in time in these exceptional fossils. And so these exceptional levels of preservation allows us to ask questions that you just can't ask anywhere else. And so for me, it's been a real focus of my research. It's really great to be up in Edinburgh now where we have a large collection of Rhynie to work with. And it goes back to that big question about how plants became more complex. So we know the early plants, like the Cooksonia ones, produce these branching stems. And we know there's also a level of complexity in the Rhynie chert, such as this plant here that has complex leaves, transport tissue, and roots. And so a real focus of the research has been trying to understand what these Rhynie plants can tell us about how plants became more complex. So my previous work, as was very kindly mentioned at the beginning, was mainly focused on the evolution of roots. We've currently got ongoing work on the evolution of transport tissues that I don't have time to tell you about today, but also really excited about. But today I'm going to be telling you a bit more about the origin and evolution of leaves. So let's just take a step back and think a little bit more big picture about what we mean by the origin of leaves. And to start with, the best place to think is, okay, so what was before leaves? So I've alluded to this earlier, but if we look at the earliest evidence of vascular plants we find, like those Cooksonia species, we can clearly see there is nothing about these plants that was leaf-like in that sense. We can see that they were just made of these branching stems and they just had terminal reproductive structures that we term sporangia at their tips. So there's no little hairy innations, there's no anything that could be thought of as leaves. And we predict that instead of having leaves, the stems themselves were the main photosynthetic organ. They were actually photosynthesizing the whole thing. Okay, so we move from this level of complexity. The next level of complexity we see is that plants go through quite an amazing innovation, which is to be able to put their reproductive structures not on their tips, but on the sides of branches. So this plant here called Nothia is able to have its stem and produce reproductive structures on the side. Now, the advantage of this for a plant is it allows you to produce many, many more reproductive structures and continue growing over a much longer period of time. It enables you to have this prolonged growth and then to continue to reproduce over a longer period of time. A pretty good innovation. But still, no leaves at all. The only lateral structures that look a bit leaf-like are these reproductive structures. And then finally, we move on to that level of complexity I showed you earlier, this plant Asteroxylon. This is a wonderful reconstruction by the artist Matt Humpage, showing the complexity of Asteroxylon. And what's kind of interesting is we see, similar to Nothria, we're actually getting the reproductive structures, these kind of like table tennis racket type structures on here. These are in lateral positions, but now there's also an entirely new organ, leaves. And so just from looking at these early fossils, we begin to see what could be interpreted as a kind of transformation series, where if we go from these plants that have these terminal reproductive structures to lateral reproductive structures, and then finally to lateral leaves and reproductive structures. But it's still very unclear how leaves evolved, how we actually undergo this switch from here to here. And it's actually also quite unclear whether this switch actually involves the modification or the change of these lateral reproductive structures, or whether leaves might be better thought of as an entirely new de novo innovation. So how are we going to go about testing that? Well, for me, the first thing to do is take a good look at the plants in the Rhynie chert. And the reason for that is that plants in the Rhynie chert actually are all the ones I've been showing you over those last few slides. And what's really unique about Rhynie is it's not just this interesting ecosystem, but it actually has a diversity of these different plants representing these three distinct levels of complexity or organization. We find in the Rhynie chert these plants that have terminal reproductive structures, we have plants that have the lateral ones, and we also have this plant Asteroxylon that has both leaves and reproductive structures. So we thought these plants in the Rhynie chert would be ideal for allowing us to investigate the hypotheses for how leaves evolved. And so what we wanted to do was to find a way of being able to contrast, experimentally to contrast the reproductive structures and the leaves. And one way we can do that is a characteristic called phyllotaxy. In botany, we use the term phyllotaxy to describe the arrangement of lateral structures on stems. So it allows us to do a side-by-side comparison between the arrangement of the reproductive structures in a plant like Nothria and the leaves and reproductive structures in a plant like Asteroxylon. So phyllotaxy is separated into a number of major types we find in living species today. You might be familiar with these, where plant leaves are often arranged in patterns such as on opposite sides of the stem, in whorls, where we get a group of leaves here separated by an area of the stem which is naked and then more leaves. And then some of the most common are actually patterns where we can see the leaves are arranged in a spiral or a helix around the side of the stem. Now what's interesting is that if we look at the diversity of plants alive today, we find that in fact spirals are by far the most common type of phyllotaxic pattern that we find present. They're found in over 80% of species that produce phyllotaxy, which is interesting. And then furthermore, we find that out of these species that develop spiral phyllotaxy, there's many of these examples that we'll be really familiar with. And it's not just in leaves that we find spirals. We also find them in reproductive structures, such as in a sunflower head or in a pine cone. And what many of you will already know is that many of these spirals have something in common. And that really comes out when we examine them and we quantify this spiral pattern in a bit more detail. So to illustrate this, let's take one example, which is how to go about quantifying the phyllotaxy of this species here, the Norfolk Island pine. So when we look down, take a bird's eye view on the growing tip of a Norfolk Island pine, we can see that the leaves are all clustered towards the very growing point. And if you took a transverse slice through that region here, you see the growing point would be right at the center, and these would be the outlines of all the leaves surrounding it. And you can see your eyes will naturally be able to start allowing you to highlight, to see and kind of recognize spirals that radiate from that central point. And in actual fact, we can recognize both anti-clockwise and clockwise spirals radiating out from the center. In the case of the anti-clockwise ones, we can see that there's 5 anti-clockwise spirals that radiate out from the center here. In the different shades of blue. By contrast, when we look at the clockwise ones, we can recognize 8. And what's interesting is this is the way that we actually quantify phyllotaxy amongst these species. We count up the number of clockwise and anti-clockwise spirals we can see, and we present that as a fraction. In this case, we just call it a 5/8 pattern of phyllotaxy. But what's interesting is that 5 and 8 are consecutive integers in the famous mathematical series, the Fibonacci series. So what's kind of fascinating is that within botanical form is written this record of the Fibonacci series is a 5-8 pattern. But I think what's most surprising is that this isn't a one-off case. The Norfolk Island pine isn't the one-off example. In fact, these Fibonacci spirals are found remarkably extensively amongst plants. All of those examples I showed you earlier are Fibonacci spirals. We have the aloe, which is a 5-8, the pine cones an 8-13, the sunflowers are 21-34. And in actual fact, if we look at that distribution of plants today that produce spiral patterns, we actually can recognize that over 91% of those spirals found amongst living species today are in fact Fibonacci spirals. So they're found, remarkably distributed, and it's been a really interesting and perplexing question for botanists and actually all sciences in general to try and work out why they're quite so common today. So what we thought to ourselves, especially with these the availability of the Rhynie chirp plants, is actually to really give some thought to when these Fibonacci spirals first evolved, whether they were present in these earliest plants, and whether they also could hold a clue for beginning to understand when leaves evolved. But to do that, what we really needed was a 3D reconstruction of these early fossils, which would then enable us to look in unprecedented detail at the structure of those leaves. And this is where this project really starts with a brilliant undergraduate student, Holly-Ann Turner, who started in my lab in Edinburgh as an undergraduate student during lockdown. So during lockdown, we needed to find a project which could be done remotely, and I happened to have some fossils which at that point in time were genuinely sitting in my house, these little thin preparations. And so during the lockdown, I was using a desktop scanner to scan in these fossils, these thin slices through the fossils. And so what I was able to do was send Holly-Ann this digital data set of these thin preparations of fossils. And then the aim for her project was to take these thin preparations and digitally stick them together to produce the 3D structure that we're familiar with. To produce the first 3D reconstruction of what an Asteroxylon stem would look like. And this is what Holly-Ann was able to do in the project. She was, for the first time, able to stick one of these fossils back together to actually visualize what it would have looked like and to actually see the arrangement of the leaves on these stems for the very first time. She was able to combine these lines of evidence with some additional transverse sections, so we were able to find evidence of remarkable fossils that preserve just that point, just that growing tip of the plant, where we can see the leaves all clustering together. And so with these lines of evidence, she could, for the first time, then actually go about trying to quantify phyllotaxy in these species. So there's two main approaches to quantify phyllotaxy. The centric approach is what I've already told you about earlier. So just to recap, we take a bird's eye view from the top, and then you look at the distribution of the leaves surrounding it. This is called the centric approach. The other major approach is called the cylindrical approach. This is where we actually try and investigate the positions of leaves as they are positioned around a cylindrical stem. And the way we do that to make it nice and easy is just to physically, if you imagine, unwrapping the stem and presenting them, the positions of those leaves, on a flat plane. And as I mentioned earlier, the next step, once you've actually done the looking and the imaging, is then to count up those spirals. And those spirals, botanically, we call peristices. And so in the case of the cylindrical approach, the spirals are actually forming helices that are winding their way around the stem, both clockwise and anti-clockwise helices. Whereas in the centric approach, you actually see them forming spirals radiating out from the center. So the way we quantify phyllotaxy is to count up those clockwise and anti-clockwise spirals using both these two approaches. And now with Holly-Ann's new data set, she could for the first time do this with a fossil. And this is the earliest time in the fossil record we've ever been able to quantify phyllotaxy in an ancient species. So she could take her digital 3D reconstruction, she could digitally unwrap it, and she could digitally slice the top of it to investigate the patterns she was finding. And what she found was there's a real prevalence of spiral patterns, and in these cases she found that both of these types, both from cutting the transverse section and when she digitally unwrapped it, were producing these 7-8 patterns of phyllotaxy. So she continued her analysis, not just with this one specimen, but all the specimens we could get our hands on. We mined through old museum collections. She was able to make multiple 3D reconstructions. And from this, we were able to find out that, in fact, this one species had quite a large diversity in the way it was arranging its leaves. There were patterns that were whorled. This is where you have a layer of leaves and then a gap and then a layer of leaves. But there was also a large number of spirals. And all of these spiral patterns had the same overall kind of description. They were what we call n to n plus 1, so 7 to 8, 4 to 5, 8 to 9. And what's very interesting about these spiral patterns, as the eagle-eye among you would have already noticed, is that in fact all of these spiral patterns are very unusual when we think about them in living species today, because these are all non-Fibonacci patterns. So although the number 8 is in the Fibonacci series, 7 isn't. And the same with all of those, the other n to n plus 1 ones I showed you on the last slide. So remarkably, the very first time in the fossil record we turn to look at phyllotaxy, we find a different type of phylotactic pattern, which is different from the vast majority of living species today. So the reason why we thought this was really exciting is it actually gave us a really new insight on the origin of leaves in this group. What it told us was that, in fact, in this early fossil plant, that phyllotaxy was really quite variable, more variable than we'd expect for many species today. It produced both whorled and spiral patterns, and we found really regular occurrences of these non-Fibonacci spirals. And what's kind of nice about this is it also made us rethink what we thought of the living species today. So previously, they'd been known, these non-Fibonacci spirals had been found in a couple of plants. And especially amongst a couple of plants known to be part of the lycophyte lineage. These had always been interpreted as oddballs, things that were doing something different, things that were likely Fibonacci to start with and had on their own lineage gone and done their own strange thing and become non-Fibonacci. But this new fossil really changed that picture, because Asteroxylon macchiae is a very early diverging member of this lycophyte lineage. And so what we're able to find from this is that, in actual fact, there appears to be non-Fibonacci spirals in Asteroxylon and in the living Lycopodiaceae you can find today, such as this plant here called Herpetia selago, which you may have seen if you've ever been walking in any of the kind of mountainous areas in the UK. So we know these non-Fibonacci spirals do exist to the present day, and based on this fossil, it seems like some of the mechanisms involved in producing the non-Fibonacci spirals were in fact, or have been, conserved over the course of 400 million years. So it's a really nice new insight on the evolution of Fibonacci spirals, and it's something we're digging into more, because it kind of throws up more questions than it answers. Because, for example, if these species are non-Fibonacci, what does that tell us about the underlying genes that are controlling them, the geometries, and so this is something we're really diving into at the moment. But importantly, it just showed us how a single fossil can really help change our perspective on leaf evolution. Okay, so it gave us this new perspective on Fibonacci's, but what about that bigger question about where leaves came from? With that, what Holly Ann needed to do was to reconstruct an axis that actually was a fertile axis and was producing those reproductive structures we call sporangia. So she found one of these in the collections and was able to reconstruct this into the first reconstruction we had of a reproductive part of this plant. In orange, we see the reproductive structures known as the sporangia, and in grey, you can see the leaves. So what we then wanted to do was to investigate the phylotactic patterns of the reproductive structures compared to the leaves. And so what she did first was plot the position of the reproductive structures on their own. And we were quite surprised about this. What we found was that there didn't appear to be any clear pattern about the position of these reproductive structures. They appeared to be, in some areas, more densely clustered than in others. You can see these three have actually been highlighted on the reconstruction, so you can see them. So this was quite unusual. There didn't seem to be any clear phylotactic pattern. But this picture began to change when we included the leaves in that picture as well. Now, all the black dots represent leaves, the orange dots are the reproductive structures. And although this has been slightly distorted, it's kind of hard to see, you can begin to see these patterns of where you can see the black dots. These are evidence of spirals again. And so what we're able to do is to carry out one of those same investigations we carried out earlier, taking that cylindrical approach. We can see the distribution of those leaves, and we find that, in fact, the reproductive structures here in orange fall into this same lattice work produced by the leaves, producing a 1516, one of these n to n plus 1 spirals. So what was so interesting about this finding is it told us that at this point in the past, there seemed to be an incredibly close developmental link between leaves and sporangia. because we're finding a single developmental program in how you produce your lateral organs was able to simultaneously produce a pattern which incorporated both the leaves and the sporangia. So this was really interesting because this was an entirely new line of evidence suggesting that in fact leaves may have actually evolved directly from sporangia. Because one explanation for why these things are so closely related, why they're in the same pattern, why they developed roughly at the same time, supports the hypothesis that in fact maybe they follow the same pattern because there was a direct modification of sporangia to give rise to leaves. This is known as the sterilization hypothesis. It suggests that you first had these sporangia, and then eventually a number of these sporangia became sterile, so stopped producing reproductive structures, and instead just produced these new organs which could be specialized for light capture. And so again, just from studying Asteroxylon in general, we're able to find new evidence in support of this hypothesis. So just to draw to a close the work on the Rhynie chert, is to say that a single fossil site has been hugely informative for our understanding of leaf evolution. The site, I'm sure, has plenty more to give in the future, and we're still investigating it in a lot more detail. But in the case of leaves, the work on Asteroxylon really gave strong support for the hypothesis that, in fact, leaves in the Lycophyte lineage evolved directly by the modification of sporangia. Okay, for the final part of the talk, I'm just going to move on, sticking with the broad theme of leaves, but this time to tell you a little bit more, a story about when ferns unfurled. And to do this, we're going to leave the Devonian world behind and instead skip forward to the Carboniferous. The Carboniferous, which gets its name because of the huge amounts of coal, also known as the Coal Age. So 300 million years ago, the UK, Europe, North America were covered by an enormous, dense, swampy forest known as the coal swamp forest. As these trees grew, they fell over into the swampy ecosystem here, and they weren't broken down. And so the carbon that had been captured from the atmosphere during the Carboniferous was buried. It was buried first in the form of peat, and then over geological time that peat became coal. And these formed the enormous amounts of coal deposits which have fueled the Industrial Revolution and also led us into our current climate crisis at the moment. So what's kind of amazing is the coal that we're burning today is actually the release of carbon which was captured by these forests 300 million years ago. But what's so interesting about these forests is they really were crucially important for us as paleontologists, because almost everywhere we find coal, we also find evidence of fossil plants. And so when I like to picture what it'd be like to walk through one of these forests, I often think of if you've ever been around the hothouse here at the Cambridge Botanic Gardens, or we have a similar one up in Edinburgh, I can imagine that it must have been something like walking through a swampy tropical hothouse. And even today, when you walk through, for example, in Cambridge or up in Edinburgh, you can actually see plants that actually wouldn't look that out of place. This is an example of a living lycophyte today, Phlegmarius. You can see its green branching stems, and then turn to the Carboniferous fossils, and again, you find something just similar. You find these large branching stems of this time of giant tree lycophytes in the past. So the vast majority of these fossils we've been finding are preserved like this. These are called compressions. You've only got this coaly remains of what the plant tissue was. But as I highlighted earlier in Rhynie, for me, the best sites are the sites with exceptional preservation. And we're incredibly lucky here in the UK to have a number of those around. And these sites are found a special type of fossil known as a coal ball. You can see why they get the name, because they're very ball-shaped. And they were discovered by coal miners, and they were a real pain to early coal miners because they typically blunt their tools on these rock-hard nodules. But I don't know how long it took, but over 100 years ago, a paleontologist realized that these nodules, when cut, actually were crammed full of plant tissue, and that plant tissue was preserved with exceptional preservation. So rather than just this coaly outline of a plant stem you can see here, when you cut one of these coal balls inside and you find a similar lycophyte stem, you can now look at it in transverse section. We can see the vascular tissue, the leaf traces, the amazing diversity of its tissues and its leaves around the outside. So again, a remarkable amount of preservation, which gives us the real chance to ask some really exciting questions about plant life 300 million years ago. And so the question we wanted to ask, and in particular, a brilliant postdoc in my lab, Dr. Raphael Cruz, wanted to ask, was when did ferns unfurl? So let's take a little step back and think about ferns in general. So I'm sure many of you are familiar with the the gradual unfurling of fern leaves. It's a wonderful sight to see in spring, and they continue all around all through the year in terms of a large tree fern and things like that. So we know that leaves develop from and gradually uncurl from a structure which is known as a fiddlehead or a crosium. They're one of the iconic features of ferns, and I'm sure you'll be familiar with seeing some of the amazing patterns of diversity. Almost every fern you'll have looked at will have these gradually uncurling leaves. But a question for us became, well, when did these structures actually evolve? And it's quite a good time to be asking this question at the moment, because the availability of next-generation sequencing data of comparative genomic analysis has really crystallized the phylogeny of ferns, the family tree of the phylogeny of ferns. So we now know that ferns really are composed of these, broadly speaking, kind of 5 key groups, and that captures a diversity of roughly 12,000 species. So they're an incredibly diverse group. And so for the first time, we could ask ourselves, when did fiddleheads evolve, and are they present amongst all major groups of plants today? So what Raphael did is he began to take that large diversity of living ferns today and break them up into smaller sort of bite-sized chunks and ask the question, are fiddleheads present? And if so, what do they look like, and developmentally, how similar are they between different groups? So what he started at first was 3 of the most early diverging lineages, the Eclositales, the Ophioglossales, and the Silotales. These lovely pictures come from the brilliant project, the Ferns of the World project, which I highly recommend you go on. You can find about the distribution of many ferns today. Okay, so we started with these 3 lineages because they're some of the most early diverging. They're also some of the most unusual. They're ones that you might not be as familiar with. The Eclositales are the horsetails. Ophioglossales, the species that grow in the UK are only tiny in size. You would easily overlook them. And the Silotales are, again, another slightly unusual group. What was interesting about all 3 of these is that from his examination, we found no evidence of fiddleheads forming in any of these groups, which is really quite interesting. Going into this, I'd always kind of assumed that fiddleheads were largely ubiquitous across all major groups of ferns. But no, there was these 3 groups where there was no evidence of them. And this was demonstrated even more by actually zooming in on the growing tips of these plants, known as the shoot apex. So this is the shoot apex of the horsetail here. You can see the leaves are forming and they're curving slightly around the tip. Same here again, and same here. But there's no evidence of that tightly coiled fiddlehead. So this was interesting because it allowed Raphael to conclude that in fact there were actually some groups of ferns that don't produce fiddleheads at all. And this suggests that there might actually be a group that are defined by fiddleheads, which he was able to recognize from continuing his analysis further out. And in this case, what he was able to find is that the Maritales and the Leptosprangiate ferns, this is where most of the living fern diversity is today. All of these major groups of ferns produce fiddleheads. And so we began to recognize that actually maybe fiddleheads were a defining feature of this group itself. And so what we got to do was to give it a kind of an informal name. We've now grouped these ferns and terming them the fiddlehead ferns, or the Cercinatophytes. And that's to highlight that we predict that actually their ancestor, like here, produced a fiddlehead, and these have been a conserved feature of the group the whole way through their evolutionary history. So the first thing is to actually recognize this as a group, and that then poses the next question, which is, okay, when did these evolve? How far can we trace these in the fossil record? And this is where we can take our dive back to the Carboniferous. Because we know those Carboniferous coal swamps were diverse. We know they had abundant ferns growing in them. And so the question became, is there evidence of fiddleheads there as well? Surely there's enough fossils that if they were there, they'd be present. So we took a deep dive into the old descriptive literature. Most of the Carboniferous work was actually described as these mines were in, while lots of the mining work was going on. And some wonderful work by Robert Kidston from 1884 described a great example of one of these fern fiddleheads. It looks remarkably similar to the living fiddleheads today. And what's great about this collection is we can obviously go back and read the early monographs on these, but the Kidston collection actually was given to the British Geological Survey. So not only can we go and read the old monographs from the 1880s, we can actually go and have a look at these, what these fossils look like in reality. So these are still the original fossils described by Kidston, illustrating the fact that there is clear fiddleheads preserved during the Carboniferous period. But one slight annoyance for us is that although we have clear evidence that these fiddleheads were there, and we've given these, they've been named as Spiropterids, as what we call a morphogenus, so you give something a name just for the actual structure of it. But we don't really know what these attach to. These isolated fragments are incredibly difficult to pinpoint down to a specific group. So again, what we really need is sites with exceptional preservation. And so we, again, trawled through the literature and found evidence from, again, the Carboniferous period, this time of France, of a remarkable example of a fossil fiddlehead of over 300 million years old. You can see, incredibly, the coral structure. And also the exceptional preservation lets us recognize key features, such as these hairs coming out the side here, which allow us to be confident in assigning this to a group of plants. In this case, to the Boturopterus plants. So this is quite amazing that we're able to find a fossil record and assign these to plants that we knew from the time. Okay, so taking those two together, what we can conclude is, first of all, the fiddlehead ferns are a thing, and they've been around for over 300 million years. And this is really exciting for us. It poses this question that fiddleheads have been there, the ferns have been unfurling for this long, but doesn't necessarily get us to the question of how these structures evolved. And so to revisit that question, let's just take ourselves on a step back to at the beginning of the talk when I was telling you about the evolution of leaves. So in the case of lycophytes, in the case of Asteroxylon I told you about earlier, there's the hypothesis that leaves evolved through the modification of reproductive structures. We've moved from these terminal reproductive structures to lateral and then leaves. However, one of the exciting things is from this common ancestor, which lacked leaves, leaves have actually had two entirely independent origins, at least amongst land plants today. The lycophytes did their own thing, and we predict, as we showed earlier, that actually they produce leaves through the modification of reproductive structures. But entirely independently from that ancestor that lacked leaves, the ferns and the rest of the seed plants split off. And there's actually a different prediction for how their leaves evolved, and that prediction is known as the telome theory. So rather than modify reproductive structures at all, what the telome theory predicts is actually you have a branching stem where it just becomes more and more complex in its branching. First of all, it produces little small side branches, then those little side branches all begin to branch in the same plane, and eventually you end up with a leaf. And this is the main theory in the literature for how fern leaves evolved. But one sort of issue with this is how do you actually go about test this hypothesis? And we were wondering whether, again, these early evidence of studying fiddleheads might give us a clue as to how we could do that. And the reason for that is if, in fact, fern leaves evolved by the modification of a stem, we might predict that leaves and stems were very similar in both their anatomy and their development. And this is the hypothesis we wanted to dive into. So we wanted to start off with a characteristic feature of the stems, which we could then go hunting for in the fossil record. So the characteristic feature of the stems we identified was that all the stems of living ferns today develop from what's called a tetrahedral apical cell. So you can see the kind of growing point has this major cell here, and it has 3 cutting faces producing new cells. And in truly exceptional cases, these features have even been preserved in fossils up to 300 million years old. So we predicted that if the telome theory is correct, that what we find, if we find an apical cell, if we find a tetrahedral apical cell in shoots, we'd expect to also find a comparable apical cell in leaves. But in the vast majority of living ferns today, in the fiddleheads of the fiddlehead ferns, this is not the case. In the vast majority of cases, their growing point of leaves is a wedge-shaped cell which has 2 cutting faces. So it's something quite different. But obviously, the vast majority of living ferns today are all recent, and so this might be a derived state within them. So what we wanted to do was actually to investigate this hypothesis using fossils, to see whether there was any evidence that, in fact, the shape of the tetrahedral cell has actually changed through time. And this is where we go to the site of exceptional preservation, back to one of these coal balls, and in this case, a coal ball collected just in Yorkshire about 100 years ago, and a number of thin sections were cut from this coal ball and were also stored in the Kidson collection, this time at the Hunterian in Glasgow. This fossil was absolutely remarkable and showed, with exceptional preservation, an amazing fiddlehead preserved from 315 million years ago. And we were excited by this fossil because it had the preservation, the cellular preservation of the tip of the apex here. But one issue we had is that if you're trying to compare a tetrahedral cell with a wedge-shaped cell, just based on a longitudinal section, you can't do it because they both just look like small triangles at the tip. So what we really needed was a way to actually capture the three-dimensional information from these fossils. So in the past, obviously, this is as far as they could have got to. This was noted as a growing point, but never properly investigated further because they didn't have the technology. But what we can do today is we can take that same fossil and we can examine it under the confocal laser scanning microscope. This is the workhorse of much of molecular biology. And what we can do is excite the fossil with a laser, and using the autofluorescence, we can actually see the structure in more detail. And so what we're able to do now is actually visualize the structure of that apical cell, not just in the longitudinal plane you can see here, but we can actually digitally predict what it would have looked like in transverse section as well. So by doing this, we could test the hypothesis of whether it was going to be a wedge-shaped cell or a tetrahedral one. The fact it has this overall triangular outline and has these positions of the predicted cell walls means that there's no way this could have been the wedge-shaped cell. Instead, it strongly supports the fact that what we're looking at here is an early example of a tetrahedral cell in the growing point of a leaf. So what was so interesting about this for us is when we put this back into this bigger picture of leaf evolution, it seems to suggest that this early fossil has its leaves actually have a lot in common with their developing shoots. So we have a tetrahedral apical cell in the shoot. We then see a switch to a tetrahedral cell in the leaves, and finally, it's a later innovation amongst living ferns today to actually have a wedge-shaped cell there. And so in conclusion, what we were able to find is this fossil allowed us to pull this all together and draw some new conclusions about the evolution of leaves in the fern lineage. First, we're able to find that likely in the ancestor of all ferns, there was a shoot apex with a tetrahedral apical cell. Now, at some point early in evolution, there must have been a switch where we got a new lateral structure formed, this new leaf-like structure, which is actually a kind of a direct copy of the tetrahedral apical cell we're finding in the shoot. Then, from this point onwards, we get the origin of fiddleheads. Only two of the living lineages today, and our amazing fossil fern Anchiopteris. And then much later in evolution, do we get this switch to the wedge-shaped cell. And then working with a brilliant botanical artist as well, we brought this fern back to life to kind of picture what it would have looked like in itself. And so this just really goes to demonstrate how using new technology on ancient fossils can begin to really give us new insights that weren't there before. Great, so with that, I'm going to wrap up. So in summary, what I really want you to take away from this is how important fossils are with exceptional preservation have been and will be in the future for changing our understanding of leaf evolution. So from a common ancestor that lacked leaves, we can see that leaves evolved at least twice independently. In the lycophytes, they likely evolved through the modification of reproductive structures. By contrast, in the ferns, we see that actually leaves likely evolved by modifications of shoots before they actually gave rise to the fiddleheads we're so familiar with today. And so with that, I just want to say that so far I've talked mostly today about the work we do investigating living and fossil plants. We also have another line of evidence in the group focusing a bit more on genetic networks. So do stay tuned, follow our lab either on Twitter or keep an eye on our lab website to find out more about what we're doing. And with that, I want to say a huge thank you to my funders. It wouldn't have been possible without their support. And a huge thank you for you listening. I'm very happy to take any questions.